Great. So like I said, today I want to talk to you a little bit about two-photon microscopy. And this was something that when tissue clearing came around and started getting popular, I was wondering if that was really going to cut into the amount of two-photon microscopy we do here at the facility. And it really hasn't. It's been interesting. Two-photon can actually help if you got a tissue that's really difficult to clear. It doesn't have to be perfect. We can put it on the two-photon and see a little deeper. We don't do a lot of live animal imaging here at the facility, but I know many of you do in your own labs. And then the other thing that's gotten really popular recently is second harmonic generation imaging. So this is a label-free technique for imaging collagen. So I'll talk about that at the very end today too. But this image here on the title slide, so this is I think you can probably figure it out. The person is holding a flashlight underneath their hand. And so that flashlight obviously emits white light out of it, which is composed of the entire visible spectrum, blue, green, and red. But you can see that only the red light is actually able to pass through this person's hand. And that's because biological tissue is not very conducive to passing visible light through it. The red gets through, but the blue and the green gets eaten up. So this is how two-photon is sometimes explained, is that, okay, well, why don't we just image in this red region or the near infrared region, and then we can penetrate so much deeper into biological tissue. But hopefully what I'm going to try and tell you today is that that's only about half of the story, and there's a lot more to it under the surface. So let's jump into this. If I can advance my slide. There we go. Okay, so let's just think about a standard wide field fluorescence microscope. So in this really bad diagram here, we have that gray objective up above. And then we've got a sample that has a bunch of green fluorophores in it. And so we're going to shine some blue light onto that sample to excite those green fluorophores. And all we really care about are the three fluorophores that are sitting on the focal plane. So the focal plane is this white line here. But you can see, because we emit a collimated beam of light out of our objective, we're illuminating everything across our focal plane, which is great, but we're also illuminating all of these fluorophores above and below the focal plane. So I think you're pretty familiar with what happens when you put a thick section onto a wide field fluorescence microscope. There's a number of sort of drawbacks to that. So one is that we have a limited imaging depth. And in wide field fluorescence, this is usually due to the buildup of this out of focus light. So the deeper you go, the more out of focus light is interfering, and you just can't image that deep into your sample. Another problem is we're exposing our entire sample to light. So this excitation light is passing from the top to the bottom. So that's going to be doing some photo bleaching to our fluorophores. And if we have a live sample, that can also cause phototoxicity. And then the biggest issue with wide field is obviously all of this out of focus light also gets collected. And if you have anything but the thinnest of sample, you get a pretty blurry image that you really can't see anything. So to mainly address this blurry image of collecting the light from all the focal planes, that's why we have confocal microscopes. So in a confocal microscope, for example here, I'm just depicting a point scanning confocal microscope. We take that excitation light and we focus it on a specific point in our sample right on the focal plane. And then what I'm not showing here is back in the detection arm of the microscope, we have a pinhole, and that pinhole is able to filter out the light that's coming from these fluorophores that are above and below the focal plane. And only the light from this one that's right on our focal plane gets back to our detector. So this cures that, or circumvents that issue of having too much out of focus light. Now we can get a nice thin optical section through our sample. We can take a bunch of those optical sections, build up a nice pretty 3D image. We still have some limitations here though. We do still have a limited imaging depth. So if you take a really big piece of tissue and I'll show you this on the next slide, and start imaging into it with a confocal, there's still a limit on how deep you can go. And again, we still have light exposure all the way through our sample. So this laser beam is passing through the top all the way through the bottom. And so again, we're doing some bleaching to those out of focus planes before we image them. And again, if it's a live sample, we have some phototoxicity there. So to some degree, 2-photon microscopes are able to address both of these problems. So let's first take a look at this idea of limited imaging depth. So just to give you an example of what I'm talking about here, let's take an uncleared mouse brain. This is a Thai-1 GFP mouse, so we should see GFP expressed in a subset of the neurons. And if we just put this underneath a confocal microscope, we drop an objective down from the top and just try imaging somewhere in the cortex here, this is what we're going to see. So this is a little movie here. Let's hopefully going to play. And what you're going to see is we're going to start from the top and start going deeper into the brain. And you can see we start to see those neurons, but pretty quickly things become quite dark and quite black. And believe me, there's still neurons there that are labeled. If you don't believe that, you can see even the background autofluorescence is fading away as well. So there's something happening here that we're not able to image very deep into that sample. And if you actually turn it on its side, it's not very deep at all. It's only about 60 microns that we've gone into that brain before we've lost all of that light. So what's going on here? What's causing this limited imaging depth? So there's two main principles that are at play here. One is absorption and the other is light scatter. So if you've heard any of my tissue clearing lectures, I've talked to you to very large depths about scatter, and we'll talk about that briefly here as well. But another issue that we have is absorption. So biological tissue has some inherent pigments in it, or you can call them molecules that are able to absorb light. So the two big ones are heme. So this is both in hemoglobin as well as in myoglobin, and then melanin as well, which we're all familiar, mainly in the skin. And what you can see is that both of these molecules have really high absorbance right here in the visible spectrum. And you can see it's highest at the blue-green side and then starts to dip down as we get to the reds and really starts to dip when we get into the infrared. So this is one of the ideas behind 2-photon microscopy is if we start using infrared light, at least for our excitation light, we can get that light deeper into the tissue, okay? Because it's not going to be absorbed by these molecules as well. However, the one thing that no one really mentions is that your fluorescence being emitted is still the same as before. So if you have a GFP molecule in there, you're still emitting green light. And that green light is a lot weaker than that laser. And that green light has to escape back out, and it's going to get absorbed by these molecules. So this is what I meant before when I said that this idea of red light passing through biological tissue better than blue or green is kind of only half the story. So we'll talk about the other half a little bit later on. And then the other issue is that not only does this light get absorbed, but this light can scatter. So whenever light interacts with matter, it can scatter, and it can scatter in two different ways. So the first way that light can scatter is in a wavelength dependent manner. And this is why the sky is blue. So we have white light that comes from the sun. It scatters off of some molecules or some temperature fluctuations in the atmosphere. And because this is wavelength dependent, we see blue, whereas the greens and the reds don't scatter quite as much. So wavelength dependent scattering happens when the molecule or object that's doing the scatter is much smaller than the wavelength of light. So this is very much on the atomic scale. And it has a very strong wavelength dependence. So the blue light's going to scatter much more than the red. And so this is one of the big reasons at play. This is why that flashlight, when you hold it under the hand, only the red light gets through, because the blue and the green are much more likely to be scattered. And if you look at this on an atomic scale, essentially what happens is we have a photon that comes in, it interacts with an atom. Its energy is momentarily, very momentarily absorbed by that atom. And then another photon is released as that atom falls, or the molecule falls back to its lower energy level. And that photon that comes out looks absolutely identical to the one that came in. The only difference is that it can go out in any direction, and this is totally random. So that's why you get that type of wavelength dependent scattering on the atomic scale. Now, we can also get wavelength independent scattering. So in this example here, we have a laser pointer. You've probably seen this in my tissue clearing lectures before. This laser pointer, the laser light can pass right through this water. We don't see anything scattering off it. If we add a little bit of coffee cream to it, coffee cream has big lipid droplets, big protein complexes, and we look at this one, now we see lots of light being scattered and coming out towards the camera, which is sitting up above this little plastic cuvette. And this doesn't matter whether I use a green laser pointer, a red one, a blue one, you're going to see this light scatter totally independent of which color I use over here. And the reason for that is these scattering centers are actually much larger than the wavelength of light, and this produces a type of scattering that's wavelength independent. And this is primarily measured by refractive index, or primarily occurs due to refractive index mismatches. So the proteins and the lipids have a much different or higher refractive index than the waterlets here. And so this is the realm of tissue clearing, is preventing this type of scatter. So 2-photon isn't going to address this type of wavelength independent scatter. So we can ignore that one for today for the most part, and you can check out some of the tissue clearing lectures if you want more information on that. Okay, great. So we've got these barriers to imaging depth, whether it's absorption, this wavelength dependent scatter, or wavelength independent scatter. So how are we going to get around these? So as far as absorption goes, I already talked to you a little bit about this. And so we'll spend a little bit more time on the wavelength scatter. But in principle, what's going to happen is both absorption and scatter are going to cause the same thing to happen in your microscope. So if we just think of a point scanning confocal microscope first, so not a 2-photon system, and again, we're trying to excite a GFP molecule somewhere in our sample, we're going to send this blue laser light in. It's going to get focused into our sample. But if that sample is highly absorbing or highly scattering, what that means is that blue light is either going to scatter or be absorbed. And that means it's not going to focus, or it's not even going to get to the focus. And both of these are going to reduce the amount of that excitation laser power that's getting to our spot here. Okay. Likewise, this green fluorescence from this molecule also is going to be scattered, or it can be absorbed in the tissue after it's admitted. And this means that if it's scattered, the light might miss the objective altogether, or it's going to be excluded by our pinhole. And also, if it's absorbed, it just never escapes the tissue. So overall, both of these things, absorption and scatter, are going to limit the amount of light that's getting back to our detector. And this is why the deeper and deeper we go into tissue, we quickly lose that signal coming back to our microscope. Okay. So how are we going to get around this? As far as the absorption goes, this is kind of the easiest one, and I already explained this to you. If we, instead of using an excitation laser in this sort of blue region for that green fluorescent molecule, if we instead use something in the infrared, and I'll tell you why we're able to do that in a second, you can see that we avoid a lot of this absorption. So the further we go out here into the infrared, the less absorption we get from the heme and the melanin. Okay. Another thing you can do, which is on the chemical sample prep side, again, tissue clearing side, is you can bleach or try and remove those pigments. Yeah. On the scatter side, we can again get around this wavelength dependent scatter by going out into this infrared region. So that's going to prevent the scatter of our excitation light. It's not going to prevent the scatter of our emission light. And as far as this wavelength independent scatter, we can equilibrate that refractive index throughout the sample. So like I said, that's tissue clearing. The other thing that we can do as far as this wavelength dependent scatter is we can reconfigure our microscope so that it does a better job of recovering that scattered light. And I'm going to talk to you about that a little bit later. But first, let's talk about how we use this infrared light to avoid absorption and avoid this wavelength dependent scatter. So this is a little energy diagram called a Jablonski diagram. You've probably seen these in our previous lunch and learns if you've been to them, or any talk that you've ever heard about fluorescence or fluorescence microscopy. And essentially what this is showing you is just the energy states of your fluorescent molecule. So this here looks like some sort of green fluorescent molecule. So we could call it GFP, or we could call it Alexa 488. And what you're used to doing in traditional fluorescence microscopy is a one photon excitation process. So we have our molecule that's sitting here in the ground state. It can absorb a blue photon, which increases its energy. It goes up to one of these excited states. And then it shakes a little bit, dances around, loses some energy, and it eventually gets down to this lowest region of this elevated energy level. And once it gets there, it can then emit a photon and fall back down. And because there's been energy lost during this dancing around up at the top, this vibration, this rotation, this photon that's emitted here has less energy than the one it absorbed. And we can see that in the color. So it absorbed blue, it released green. Green has less energy. Green light has less energy than blue light. Okay, so this is what we're used to doing. But there's another way to move through this cycle that works with red photons instead of blue photons. And I think you can probably guess that if you take two red photons that combined have the same energy as this blue photon, and if they hit your fluorophore at exactly the same time, they can both be absorbed, creating enough energy to get up to this excited state. And then the second half of the process is exactly the same. We do this rotation and vibration dance down to the lowest point of this excited energy state. And then we fall back down to the ground state, releasing a green photon. So the only difference in two photon microscopy in this whole fluorescence energy loop is that instead of using a single blue photon, we're using two red photons, which combined have the exact same energy as this blue photon. All right. This is a bit of an oversimplification, but these energy levels don't look exactly the same for two photon as they do for one photon. But this is sort of the best way to understand it. Okay. So this was originally proposed by Maria Meyer. This was in her doctoral thesis back in 1930, and then she ended up going on to win the Nobel Prize years later for something totally different in physics. And it wasn't actually until the 60s that this was, I think it was after her Nobel Prize, that this was actually proven to be viable, this idea that you could excite fluorescence with two photons of a lower energy. And then after that, it was still another 30 years before anyone put this into a microscope. So this was the two photon microscope was originally developed by Winfried Dank and Watt Webb at Cornell. And this is what it looks like. So This is the diagram of a confocal microscope that I showed you earlier. And there's two big changes to turn a confocal microscope into a 2-photon microscope. And the first and most obvious one is on the excitation light path. So I told you we're not going to use this blue light for excitation anymore. We're going to use red light or near infrared light. And that needs to have some pretty special properties to it for this to actually work. So let's take a look into this excitation. So I think everyone's used to seeing these diagrams for one excitation of a fluorophore. So here we have, this is GFP. Here's its absorption in this blue color, and here's its emission in the green. And there's a couple measurements that we usually do on these fluorophores. One of them is the extinction coefficient over here. And the extinction coefficient is the probability that that molecule is going to absorb a photon of blue light when it's hit by it. And the higher this extinction coefficient is, more likely it is to absorb, and probably the better the fluorophore is. And this is a linear process in 1-photon microscopy. So what I mean by that is if you double your laser intensity, you will double your fluorescence intensity when you're on the microscope. Up until a certain point, there's obviously a saturation that you reach. But this is totally linear in 1-photon microscopy. Not at all in 2-photon microscopy. So in 2-photon microscopy, instead of these really nice, clean absorption curves like we have here that have an obvious peak, these are a couple equivalent absorption curves. So this is Alexa 488 up here in the light green. That's its absorption. And this is its 2-photon absorption, is this red line here. And you can see it's just kind of crazy. It goes all over the place. It spans 300 nanometers of spectrum. And so this starts to become a little messier when you're trying to plan out a 2-photon experiment. And the other thing is that this is now a non-linear process. You need 2 photons to hit that molecule at the same time. And so what that means is that your fluorescence intensity actually scales with the laser intensity squared. So it's no longer linear. It's now non-linear. And the probability of those 2 photons hitting one fluorophore at exactly the same time is incredibly low. This is very, very low. If you tried to measure an extinction coefficient on it, it would be nowhere near the same realm as what you have for 1 photon. And in fact, you usually need about 1 million times more photons than we do in a 1-photon process. So the only way we can do this is with a very specialized laser. And again, this was a Nobel Prize worthy discovery by Moreau and Strickland. They got the Nobel Prize for this a few years ago for their method of generating high intensity, ultra short optical pulses. Ashkin was also on this Nobel Prize. He got it for optical tweezers, which is really cool. Not related to what we're talking about today, but if you just want to look at something that's really neat, you can move objects, microscopic objects around with a laser beam. It's really cool. So what these lasers that we use now for 2-photon microscopy are, they're infrared pulsed lasers. And I'll explain a little bit more to you about what that means on the next slide. This is the type of laser that's been traditionally used. This is called the Ti-sapphire laser. And so what that means is somewhere inside this big box, there's a sapphire crystal that's been doped with titanium. And if you shoot a green laser at that titanium crystal, that titanium sapphire crystal, you can see we have this pretty broad absorption spectrum for that crystal. And then it emits a lot of infrared light, okay, over a pretty wide range. So this is what's happening inside this box. We have a green laser that pumps that crystal, and then we get out a lot of infrared light from it. There's a lot more complex optics in here that allow you to select certain wavelengths across here so that you're not getting this whole spectrum all at once. You can tune it to specific wavelengths. And then we also have some optics in there for creating pulses of light, which I'll talk to you about in the next slide. These are really powerful. So your average power here is over 2 watts. And this is what's been traditionally used for 2-photon microscopy. Now, more recently, this is the actual laser that we have on our system here in the HCBI. And they use a slightly different technology for this, which allows a much broader tuning range. So now we can tune out to 1300, whereas the Thai Sapphire is limited to about 1000. And this is really handy for doing 3-photon excitation or triple harmonic generation, or also just doing 2-photon of far red dyes. So this is a really great technology that we were able to add a couple years ago that's sort of expanded the palette for 2-photon or multi-photon work. So I said that these lasers were pulsed. So why do we need a pulsed laser? So if we think about what's going on here, what we need to do is we need to pack as many photons as we possibly can into our laser beam that we're scanning back and forth across our sample. Because I said to have that probability of a 2-photon event, you just need an incredibly large number of photons there. So our microscope's doing some of this work for us. Our microscope, any point scanning designed microscope, does a really good job of spatially confining these photons. So if we look at our laser beam that comes into our objective lens and then gets focused into our sample, if you draw a line anywhere along here, you should hit about the same number of photons, right? But you can see that once you get down to your beam waist here, you hit that same number of photons in a very, very small space. So let's say we're hitting 3 photons with each of these lines, but up here that's spread across the entire back aperture of the objective. Here it's focused to a tiny spot, let's a couple hundred nanometers in size. So our microscope helps us out a little bit here by doing this spatial confinement. But the problem is if we took a laser, the usual lasers that we use are what are called continuous wave lasers. So they're always pumping out an equal number of photons. If we took a continuous wave laser that put out enough photons that combined with the spatial confinement gave us a 2-photon effect here, we would actually totally cook our sample. So if you had any sort of biological tissue there, you would just light it on fire and melt it immediately. So this is not an option for doing 2-photon microscopy on biological samples. So what we need are these pulsed lasers. And what the pulse laser does is it combines our photons in time. So what that means is it packs a whole bunch of photons into a little pulse. And then we actually have this big break where there's no photons at all. And then we get another pulse with a whole bunch of them packed in. And this means that we have this incredibly high photon density because we've crammed a bunch of photons in time and also in space. But the average power, the amount of light that's actually hitting our sample on average is quite low. It's in the range of 1 to 10 milliwatts because the sample actually sees this laser as being off more than it's on, okay? So hopefully that makes sense. So we're packing a whole bunch of photons into a really short period of time into a really small space, but we only hit it for a short period of time with those photons so that we're not cooking our sample. Now, one of the drawbacks to trying to pack photons into time like this is that those photons aren't pure. So it's impossible to make a really short pulse with photons of one specific wavelength. So if you were to take a look at our laser, if we tuned our 2-photon laser to 800 nanometers and you took a look at the light that was coming out of that laser beam, it actually has frequency components of wavelengths from about 750 to 850, okay? And it all comes out in a single pulse, but anytime it hits an optical component, any piece of glass or a mirror, because there's a refractive index mismatch there, it's going to spread out this pulse. So these frequency components are going to shift away from each other. Our pulse is going to get longer, and we're not going to have that same temporal confinement of our photons. And so this is what we call dispersion. So we're spreading out these frequency components. And that means that when we have less temporal compaction, when we actually get to our sample, there aren't enough photons there anymore to create the two-photon effect. So this can be a problem. And now what we have on our lasers, and we have this on the system here at the HCBI, is something called a pre-chirp. And one way to do this is to take your original pulse, this original pulse that's coming out of your laser, you pass it through a series of prisms, and that actually stretches it out in the reverse way. So you can see here we had our our high frequency blue light was stretched to the left, and our low frequency red light was stretched to the right. If we pass these through these prisms, we actually do the opposite. So we get the red light to the left and the blue light to the right. And then when it goes through all the optical elements of our microscope, due to that dispersion, they get realigned back on top of each other. So this is a way of artificially spreading out that pulse as it comes out of our laser head. We let it go through our microscope so that by the time it gets to your sample, it's realigned here. And this is a setting that we can show you if you want to see it when you're here. It's actually just a really easy slider. Let you slide back and forth, and you just watch your sample to see when your signal gets the brightest. It's a pretty easy setting to take care of. Okay, so we talked about using infrared light to circumvent absorption and to circumvent this wavelength dependent scatter. And another way that we can try and circumvent this scatter is to do a better job of recovering all of that scattered light. So this is the second half of a two-photon microscope. And in my opinion, this is actually the most important part of a two-photon microscope. So yes, this infrared light can penetrate much deeper into tissue, which is fantastic. But as I've said multiple times already, we still have this green light that has to get back out, and it's going to get absorbed. It's going to scatter. So what I think is the magic behind a two-photon microscope is with a traditional confocal, we take that emission light, it comes back, and it has to go through this pinhole to filter out the light that's coming from above and below the filter plane, or above and below the focal plane. And this pinhole actually excludes a ton of light. This is a really inefficient process. And so with two-photon, we're actually able to get rid of this pinhole, okay? And what we actually do is we move our detector forward into the light path of the microscope. And I'll tell you in a second why we're able to do this. But this is what it looks like. So this is the two-photon system that we have in the HCBI here. And so this is a traditional confocal scan head on the back. You can send the light back there and try and get it to the detector, but it's much more efficient if we use this detector that's mounted here really nice and close to our objective. So what happens is our infrared light comes in, gets focused into our sample. We get our fluorescence emission that comes up. We put a mirror right here that bounces that emitted light straight over to this detector. And that allows us to be incredibly efficient, have very few pieces of glass, and collect that light really quickly. So why do we not need a pinhole? So this is just showing you again, what I showed you earlier is that with this confocal microscope, usually we excite all these fluorophores above and below, and then filter out that light at the pinhole. But with a two-photon, that's not the case. And this has two great effects. One, it helps us to detect light more efficiently, but we're also not going to be bleaching throughout our sample anymore. And the reason for that comes back to this non-linearity that I was telling you about. So when I talk to you about one photon excitation scaling linearly with the concentration of photons, If we reduce our laser power in half, we reduce our fluorescence in half. But that's not the case with 2-photon because we're nonlinear and we're scaling with that photon flux squared. If we cut our laser power in half, we actually cut our fluorescence by 1/4. And so you're saying, why would you ever want to cut your fluorescence? You don't. But if we look at our microscope, so in 1 photon here, we focus that blue light in to excite these green molecules. And it doesn't matter where that green molecule is, it gets hit with quite a bit of blue light and enough to excite it, okay? But yes, that blue light is the most concentrated here at the middle, and it's slowly getting less and less the further we go up. But we're still able to excite those fluorophores pretty far above and below our focal plane. With 2-photon, that's not the case at all because we have this nonlinear fall off of our excitation ability. We really can only excite the fluorophores that are right here at the focal plane, or we really only have a high probability of exciting right at that focal plane. So that means these fluorophores that are even just a little bit outside of our focal plane, the ability to excite them is falling off so quickly that we just don't. So because we know that our fluorescence is only occurring on this focal plane right here, we don't need a pinhole anymore to filter out light coming from these ones. And even if the light from here scattered bounces off a bunch of different places in the tissue, as long as it gets into the objective, we knew that it came from our focal plane. So what we're trying to do is just make the most efficient light path possible now in a 2-photon microscope and collect all of these photons. Anything that gets into the objective, we know it came from precisely this position. Yeah. And so I think this is kind of the most famous photo. There's various versions of this in 2-photon microscopy, but this is just what I was trying to show you in that diagram on the last page. So this is a cuvette, and inside this cuvette is a green dye. It's probably fit C. And if you shine blue light on in a 1-photon excitation process, you can see you're exciting that fluorophore from the very top all the way to the bottom here. Okay, so here's your focal point in here, but everything above and below it is getting excited. If you do this with a 2-photon process, so the only difference between these two is that over here we're using this infrared light to excite. And you can see, it might be hard to see on your screen, but there's a tiny little dot right there at the end of my cursor. And that's the only spot where that green dye is actually being fluoresced, all right? Or being excited. So we don't have any emission above or below that. And that's actually about 1 femtoliter. It's a tiny, tiny little volume. So that's why I'm actually just going to go back here. Sorry. So that's why we can move this detector right next to our objective. And by moving that detector there, any light that comes from our focal plane here is going to get collected on that detector. We're not going to lose it by going through additional optics here. And we're able to assign that to our final image. And that is, in my opinion, the real magic behind 2-photon. It's just a much better, much more efficient collection of light. So how deep can you go with a 2-photon microscope? It ends up, there is actually still a theoretical depth limit to it. In general, you're never going to reach this theoretical depth limit. I've never reached it. It's still going to be very dependent on your tissue, how much pigment there is in that tissue, how many refractive index mismatches there are in that sample. There's still a lot of other things that are going to affect how deep that you can go. But if you just look at theory, although I told you that the highest probability of a 2-photon event is right here at the waist of this beam, there is still a very low probability of it happening elsewhere. And what that means is as you image deeper and deeper into your sample, you get a larger and larger volume of this hourglass shaped light beam that's coming out of our laser. And so at some point, you are going to be exciting a few fluorophores outside of that focal point. And also the light that's coming from this focal point is going to get scattered on its way back or absorbed and not make it to your objective. So at some point, the amount of light being excited outside of your focal plane is going to become equivalent to the little tiny bit of light that's getting back from your focal plane to your objective. Theoretically, this happens at about 1 millimeter. So in practice, I think the best I've ever done on our system was about 600 to 800 microns. That was into a really bright sample in a zebrafish brain, I think it was, which doesn't scatter all that much. So yeah, that's your theoretical limit, but practically you're usually a lot less than one millimeter. So just a couple other practical things to keep in mind for 2-photon. Multicolor experiments can become pretty difficult. So I told you the absorption spectra for 2-photon is not at all equivalent to 1-photon. I think a lot of people often come in and say, okay, I'm going to do 2-photon. My dye absorbs 500 nanometers. That's its peak for 1-photon. So I just double that for 2-photon, right? It ends up that's not correct at all. And it's because those energy levels end up being different for 1-photon and 2-photon excitation. There's some great resources out now. This is a paper, I think this is from Luke Lavis's group at Genelia, which is awesome. They published these 2-photon absorption curves for most of the most popular dyes, which are great. But if you take 3 dyes here that we commonly use in the facility, so Alexa 488, 568, and 633, if you look at their emission spectrum, they're pretty well separated. So with one of our normal confocal microscopes, we'd have no problem imaging these 3 dyes individually. We'd use 3 different lasers to excite them. We'd have no problem separating them spectrally. But when it comes to 2-photon, so Alexa 633 is the green curve, 568 is this red curve, and 488 is the red curve here. You can see the optimal place to excite all 3 of those dyes is right around here. It's just a little under 800 nanometers. So somewhere between like 770 and 800 is the optimal wavelength for exciting all 3 of those dyes. So if you have those 3 dyes in your sample, you're going to tune your 2-photon laser to about 780. All of them are going to get excited at the same time. So now it's going to be a little more difficult to separate them. The 488 and the 568 aren't going to be too bad, but you're going to have a tougher time with this 568 and the 633 because you can see there is a fair bit of overlap between these. And just to compound that a little bit more, with 2-photon, when we're using this detector out on the side here, we call this a non-descanned detector. This is a filter-based separation. So we have optical filters that are inside that detector. We don't have, if you've been using our 880s or 980s or whatever 80s that we have now, you can finely tune your emission spectrum very easily to almost like 3 nanometers. But in a filter-based system, you can't do that. And on top of that, the one that we have at the moment actually only has 2 detectors in it. So right now, you would have all that light from those 3 different dyes coming back here onto this non-descanned detector, and you could really only image 2 of those dyes here. So what we'd have to do in this situation is we couldn't use this non-descanned detector. We'd have to go back into the confocal scan head here. The good news is we can open up the pinhole, so that doesn't become a problem, but there's just a lot more glass to pass through here. So your signal is not going to be as great going back there, but we're still able to do it. So this is sort of just like a note of caution, let multicolor experiments with 2-photon become a lot more difficult. Cool. Okay, so that's 2-photon. It solves a lot of problems. We get fluorescence only coming from our focal plane. We don't get bleaching outside of the focal plane, and we have a nice large imaging depth. Still a really slow process. We've got to scan that laser back and forth 1 pixel at a time. but good news, we can put up with that. So 2-photon, we're just really scratching the surface here. So this is something, a field called nonlinear optics, and there's 2-photon excitation, there's 3-photon excitation processes. We can do second harmonic generation, third harmonic generation. There's a lot of specialty sort of niche imaging methods that exist in this world of nonlinear optics. But in the last like 5 minutes, real quickly here, I just want to talk a little bit about second harmonic generation, because this is something I've seen a lot of people doing recently. So second harmonic generation, this is not a fluorescence process, but it's something similar. And this is often seen in certain crystals, and that's where most of the research in this area has gone on. But interestingly, collagen, a biological molecule, collagen, acts very much like a crystal. It has a very repetitive structure to it, and it can perform something called second harmonic generation. And what that means is if you send in 2 photons of infrared light, so at 900 nanometers, collagen is able to combine the energy of those 2 photons and spit out another photon with double the energy. So a photon comes out at 450 nanometers. And this is a very exact process. Like I said, this is not fluorescence. So whatever light you put in, the light that comes out is exactly half the wavelength or double the frequency. This is often called frequency doubling. So this is a label-free imaging technique. You don't have to put any dyes or anything on your collagen. If your collagen's there, it's going to do this. Those 2 identical photons are combined to create one higher energy photon, exactly half the wavelength. And this can occur in collagen, mainly in type 1 and type 2. And the only trick to this is that the wavelength of this incoming light here has to match somewhat closely to the atomic size of the crystal structure. So for collagen, we usually find this works best between 900 and 950 nanometers. Okay. And so what that allows you to do is, this is just an interesting sample. This is from a local biotech company here who makes purified collagen sheets, and they gave me a piece of this. So you can build up these incredible 3-dimensional images without any actual labels whatsoever. So this is something that we've had a lot of people doing recently, and I just wanted to give you a quick introduction to it before the very end here. Cool. So yeah, just in summary, absorption and light scatter can limit our imaging depth in fluorescence microscopy, and we can reduce these when we use infrared excitation light. And we can use 2 photons of infrared light to excite our fluorophores in a similar manner to what we do with 1 photon. And also we can remove the pinhole in our microscope and use a non-descanned detector to increase the signal. So these are the really 2 take-home concepts today. It's just this idea of using 2 photons of infrared light, which have the same energy as 1 photon of visible light, and getting rid of that pinhole and being much more efficient on the detector side of things to increase your signal. Cool. So thanks for listening. I'm going to end our recording here.